Okay, so we are going to start by talking about carbohydrates. This is our first class of biomolecules that we're going to look at. So what we need to do is look at the structure of our simple sugars. And these are going to be 3 to 6 carbons large. And then we'll talk about our polymers of carbohydrates, which are called polysaccharides. And we're going to talk about how their form follows their function. Okay. So carbohydrates, these are some organic molecules, right? Some biomolecules that can either be a chain of carbons like this, or we can have a ring of carbons, something like this. And the reason that we call them carbohydrates is because they are a carbon atom with the parts of water. And so we're going to use the general formula of carbon plus the parts of water to figure out the chemical formula of any carbohydrate that we're interested in. And they have this general formula of CH2O times N, where N is equal to the number of carbon atoms. Okay, so we have our 6 carbons. So therefore, if N is equal to 6, we can figure out the chemical formula by just taking this and plugging it in. So if N is 6, that means that we're going to deliver those 6 carbons to, and then we're going to do 2 times 6 is going to give us 12 hydrogens. And then this N is 6. We're going to get that to our oxygens. So our glucose has the chemical formula of 6 carbons, 12 hydrogens, and 6 oxygens. So we call this C6H12O6. Okay. And our carbohydrates are can include sugars, starches. We'll talk about glycogen. We'll talk about cellulose. We'll talk about some other ones as we come along here. So our monomers for this class of carbohydrates are called monosaccharides. Mono means one, saccharide means sugar in the Greek. So monosaccharide literally means one sugar. Okay, this is a simple sugar. We can't break it down into smaller sugar molecules. And this is what we're going to use for our immediate source of cells, specifically our friend glucose. And we can also use these as building blocks for other types of our organic molecules that we're going to talk about. And we can talk a little bit here about our friend's ribose, which is a 5-carbon sugar. So anytime we see a little bend in this chemical structure, even though there's not a C there, if there's a bend there, there's a carbon. Okay, so we have this 5-carbon sugar of ribose. We have another 5-carbon sugar of deoxyribose. These are important building blocks that are going to help us build DNA, and we'll talk a little bit more about that later in this chapter. Okay. So our monomers, they're usually between 3 and 6 carbons. Once we start getting more than 6 carbons, then we kind of start to delve into the realm of other disaccharides or polysaccharides. Okay, but we stop and talk about this thing called isomers. Okay, we've already decided that glucose has the chemical structure of C6H12O6. And if we count all these carbons up, we're gonna find that we have 6 of them. If we counted up the oxygens, we'd find 6. If we count up the hydrogens, we'd find 12 hydrogens. Okay, but fructose, let's count up our carbons. 1, 2, 3, 4, 5, 6. Okay, all right, let's go C6 down here. All right, let's look at our hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 of those. Uh-oh. Okay, what about oxygens? Okay, so 1, 2, 3, 4, 5, 6. Aha! So fructose has the same chemical formula as glucose, and you could do this on your own, but galactose also has the chemical formula of C6H12O6. But what we're going to see, if we look at these closely, and I'm just going to erase this for now. What we see here is that they have all the same number of atoms, but those atoms are put together in different ways. So the difference between fructose and glucose is where this double bonded oxygen is on these carbons. So if we're going to count these, we count 1, 2, 3, 6. So the double bonded oxygen is on the 5th carbon. On our glucose, we see that our double bonded oxygen is on our 6th carbon. Okay, this is a structural isomer. They're going to have very different structures. They're going to function in the body differently. Okay, so that's a structural isomer. Iso means, let me write that up here. Iso means same. Okay, we saw that when we were talking about isotopes. Okay, so it's the same root. All right, so let's talk about the difference between glucose and galactose. Okay, this one's a little bit trickier. We see that the double bonded oxygen is still on carbon 6 up there, but what we'll notice on glucose, our OH group is on the right-hand side, whereas, oops, let me put that up. Let me put that in a different spot. But in our galactose, the OH group is on the left-hand side. So that's going to change how these different monomers are going to hook together and change how they build dimers and polymers. All right, but let's spend a little bit more time talking about glucose because this is the rock star of the biological world. Okay, so we can have this chain form that we just looked at. We can also have this as a ring form, and when glucose is in an aqueous environment, all right, you guys are seeing my dyslexia and my lack of confidence and ability to spell. Okay, but when it's in an aqueous environment like our cells, it's going to form this ring shape. Okay, so this is where 99% of our glucose molecules in our bodies are in this, and then 1% are in that chain form. Okay, now when glucose goes from being a chain to a ring, the location of like how we're going to change that form, we can either have this alpha version where the OH group is pointing down, or we can have the beta version, this funny little B for beta, of where that OH is sticking up. Okay, and the reason that this matters to us is because if the glucose molecules are in the alpha form, that is going to, well, let me just go back and say it a different way. If the glucose ring is either an alpha form or beta form, they're going to form these big long chains of polymers differently. Okay, so in our alpha form, we have the oxygens hooking these monomers together, these monosaccharides together in a way that that these are called alpha bonds because that's in the alpha position. They're easy to break. Most organisms have the enzymes to break that chemical bond and release this glucose molecule so that it can then be available for cellular respiration and obtaining energy from. Okay. But if we have a bunch of glucose molecules that are in their beta form, this bond here, but almost all organisms lack the enzyme to break that beta bond, meaning that these glucose molecules here are not available to us as a source of energy. And so we often see these beta bonds being a part of the structural components of organisms. And we're going to get it, we'll talk a little bit more about that here in a second. Whereas our alpha bonds, these are easy to break. And so we see this as a form of energy storage. Okay. So dimers, these are two monosaccharides that are hooked together. So di means two. Okay, mers, we know what those are the parts of. So a disaccharide means two sugars. Okay. And usually what they use, the function of our disaccharides is for the transport of glucose molecules. If we have just our glucose all by itself, it's going to get snagged up by an organelle that does cellular respiration, but we need to move it from this part of the body to that part of the body. So we're going to hook it with something else so that we can move it around. Okay, so our friend sucrose, which is we know as table sugar. Okay, this is the stuff that you might put in your coffee or on your cereal or make Kool-Aid out of. Okay, this is a glucose molecule linked with a fructose molecule, and we can see that chemical structure right there. Okay. This is what plants are going to convert their glucose that they make through photosynthesis and turn it into sucrose so they can get it from the leaf down into the other parts of the plant where it needs to be used. All right, so some other kind of important disaccharides that we can talk about is lactose. This is known as milk sugar. Okay. And if we remember the types of organisms that drink milk and use milk as their food source, those are going to be baby mammals. Oh, I'm running out of room. Okay, baby mammals. Okay, this is a glucose molecule linked to this other thing called the galactose. Okay, and you guys do not have to memorize the chemical structures of these. You need to know that these are, you need to know the formula, but you don't have to be able to draw me this little weird shape. Not for this particular, not here. I'll tell you when. Okay, it's very rare, so I'll let you know. All right, so the bond between glucose and galactose is one of these beta bonds. And this B is the Greek letter beta. It looks like a fancy B. Okay. So if you are lactose intolerant, you lack the enzymes to break this beta bond as an adult. So congratulations. If you are not lactose intolerant, that means that you, welcome to the X-Men, you are a mutant. Where you are, as an adult, you can still produce the enzyme called lactase to break down the sugar lactose as an adult. So that's what you can do with genetic mutations. You can't actually control magnetism or endobrand. Okay, maltose is another important one. You probably heard of this as malt sugar. These are two glucose molecules hooked together in an alpha bond. So I'll just write a letter A there for alpha. We often see this in plants, especially in seeds that are germinating. Because now we can get these two glucose molecules, break one bond, and have double the amount of glucose without having to spend more energy to modify galactose or fructose to turn it into glucose. Okay, so there is our dimers, our disaccharides. Our polysaccharides then, right? We know what poly means. Poly means many. Okay, so these are many mers. So a polysaccharide are means many sugars. Okay, where we are going to have long chains of monosaccharides that are linked together to form this long chain. These long chains can be branched or unbranched. And we'll look at a couple examples of that. We're going to use these for a few purposes. We can use them for energy storage, okay, for our mid to long term energy storage. Again, we might hook those up as dimers, or we might put them into this long chain of polymers. Okay. We can also use these for structure. This is especially true for organisms that have cell walls. We'll talk about some of those in just a second. Okay, so some examples of polymer or polysaccharides. This is glycogen. This is found in animals as an energy storage. Starch is found in plants as energy storage. Cellulose, plants as structure, and chitin, this is in fungi, and arthropods. So like insects, spiders, crabs, and stuff as their exoskeletons. So this word is pronounced chitin, not chitin. I know Greek did that. We can blame Greek. Okay, so we have all these, let's call these glucose for now. All these glucose molecules that are hooked together in these bonds. We can use our hydrolysis reactions to break them apart into our component monomers. All right, so let's talk about starch and glycogen. These are our energy storage. Okay, starch is going to be in plants, and glycogen is going to be in the muscle cells of animals. Okay, so starch, it's hundreds of these alpha glucose molecules that are in these big long chains. They can just be stacked end to end to end to end. This is kind of like in foil, so that's why it kind of looks funky. We're just going to learn the name of starch. But if you want to be an extra nerd, the amylose is the unbranched. And then amyloipectin is the branched form of starch that we see in animals. And again, we're gonna pack these glucose molecules end to end to end to end using that easy alpha bond. You can break them apart. Plants usually store their starch in their roots in these underground. That's why potatoes are so delicious, is because they're full of starch and full of these glucose molecules that we can just break apart and then use them for producing cellular respiration. Okay. Glycogen is often called animal starch. This is another glucose in our alpha bonds. But these are usually more branched than what we see in plants. And if you've ever heard of like marathoners hitting the wall, that's because they've depleted their sources of glycogen and they have to go and use their fat reserves. So they've been breaking down all of these molecules using those glucose molecules to produce energy. And now they don't have that storage anymore, so they have to go and use their fat reserves, which is actually a little bit harder and a little bit less effective of the reason. And again, we'll talk a little bit more about that when we get to the cell respiration chapter later in the semester. All right, so cellulose. These are polymers, right? So they're still glucose molecules. Okay, but now they're hooked together in those crazy beta bonds that are hard to break. Beta bond, hard, no break. Okay. And that means that most organisms do not have the enzymes to break this bond. So that's why these are used in the structural components in plant cells. So when we get to talk about, so the plant cell walls have a lot of cellulose. So I like to use this analogy of the plant cell walls being the door frame. And when I push up against it, or at least when I was a little kid, I used to be able to climb up the door frame by pressing up against it. And that's kind of how the cell wall works. And because this is nice and rigid, we can have, or when we have a lot of water in the plant cells, that's going to push against that cell wall and it's going to keep your plant upright. When you forget to water your plants, they get all droopy and dramatic. And that's because they don't have enough water pushing up against that structural support of the plant cell wall. Chitin has a very similar structure as cellulose. We have all these glucose molecules that are being held together in our beta bonds. But the thing that makes them different is this nitrogen containing functional group that modifies those glucose molecules. And again, we see these in the exoskeletons of arthropods, right? The crunchy bit that makes them hard. We also see them in the cell walls of fungi that again act kind of as that structural support for the fungal cells, similar to what we saw with the plants. Your challenge question is going to be, how do the structure of starch, glycogen, and cellulose affect how they function? So when you answer this question, I need you to describe the structure of starch and the function of starch, the structure of glycogen and the function of glycogen, and then the structure of cellulose and the function of cellulose. So make sure that you are hitting all those parts. Okay.